We looked at mitosis and the phases in cell division. The process of meiosis, we're going to see how it's a little different. It is cell division, but we're making eggs and sperm. So production of gametes is what it is. And the names of the phases are really the same as for mitosis. However, there's two divisions in meiosis. It's divided twice. The cell's divided twice. So when you're divided twice, you're going to end up getting half the DNA as the original cell. So really the first part is phase 1. And that's made of interphase, prophase, metaphase, anaphase, telophase. And then there's phase 2, which is made up of interphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2. It includes interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, say even 1. Interphase 1, we're starting off with chromatin. So this is the form of DNA that looks like a bowl of spaghetti, so it's just loosely packed DNA. Prophase 1 now, we're going to form chromosomes. Chromosomes in meiosis, in prophase 1, they form pairs. All right, so they pair up. And sometimes when they pair up, they exchange parts of their DNA. So I'm just going to look at these two chromosomes. Now, if you have licorice at home, if you have red and black licorice, this would be awesome. So what you could do is take 2 pieces of black licorice, make an X, either tie them together with string, or I used to use like a marshmallow in the middle. Okay, so let's say I'm going to put marshmallow, and then this one, same with red licorice. All right? So when they're paired up like this, sometimes they exchange pieces. So if you're demonstrating this with your licorice, you're going to cut a piece off of one, or you're going to cut pieces off of each one and exchange them. Say right here, this part, they overlap, and they exchange pieces. So right here, we can see this piece from this chromosome is exchanged for this piece. So now, notice that this is mixing genes right here. This is because, remember, the chromosomes contain the genes. They contain DNA. So when there's an exchange, this is shuffling. It's like shuffling a deck of cards. And this is just in the process of meiosis, the genes are mixing. So this is way before fertilization. So this is called crossing over. So this process, DNA is exchanged between chromosomes. That's what it is. So DNA is exchanged. You can see it right here. And if you have licorice, then you cut it and exchange the pieces of licorice. That's crossing over. This is mixing genes. And this can happen during this time because they're paired up. Okay, metaphase now, middle. Think meta, middle. So now these chromosomes are going to line up, but they're going to line up in pairs. These pairs are called homologous chromosomes. They code for the same traits. They might not be, they're not going to have identical genes because one comes from mother and one comes from father. But they code for the same traits. So if we think of like the color of dog breeds, their color, the coloration. If there's a gene for that merle color, let's say, on this part of this X, this red one, then there could be a gene for white or black color on this one, or maybe even merle. Might be the same in that case. All right, but one of these came from mother, one came from father, but they contain genes for the same trait, not exactly the same genes. Okay, so remember, a trait is something you inherit. It's a characteristic that is inherited. In other words, it's passed on from your parents. It's in your genes. Okay, so here we have, these line up now, and they're also called tetrads. And you're wondering why tetrads, because tetra means 4. about it, there's really 4 halves of the chromosomes here. So you have 1, 2, 3, 4. So each half is called a chromatid, and you have 4 chromatids, so it's called a tetrad. So it's a tetrad. So now we have them lined up. You'll notice this is different than mitosis, because mitosis, we didn't have pairs. We did not have homologous chromosomes lined up. There was no crossing over in mitosis. There's no mixing of genes in mitosis. So now, metaphase, they line up. Anaphase, now that we're going to separate the entire chromosomes. Chromosomes are separated. Okay, I'm not writing a description of each one of these, I'm saying it, you can write that. All right, telophase, remember, is formation of 2 new nuclei. Okay, so over here, one cell is going to have 2 black X's, 2 black chromosomes, and the other cell is going to have 2 red chromosomes. Now, if there was crossing over, we'd even have more shuffling of DNA. So I'm going to show that, actually. Let me show one of these crossed over. So we're going to say this one has a piece of red, and this one has a piece of black. Cytokinesis, then, is the splitting the cytoplasm. So, well, this is the first half of meiosis, which is made of these phases: interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1. The next thing that's going to happen is each of these cells are going to divide. Okay, so this one's going to divide into 2, and this one's going to divide into 2. And the second half of meiosis is really the same as mitosis. Meiosis 2, we just use Roman numerals. All right, so now we have these 2 cells that were produced from meiosis 1. They're each going to divide into 2 cells, right? So the first thing then, this is pretty much just like mitosis now. We're going to go through prophase 2. The nucleus starts to disappear, and chromosomes are formed. Now here I showed the chromosomes already. So really, in interphase, that's when the chromosomes are not, they're not visible yet. It's really, it's chromatin. It's when it's all, the DNA is like a bowl of spaghetti. Next, we have metaphase. Okay, so we're going to line these up. And remember, we had crossing over happen, so I'm showing that here. Okay? Next, we have anaphase 2. So anaphase is when we start pulling the chromosomes apart. Okay? Then we have telophase 2. And this is when the nuclei are going to be formed. And remember, we had crossing over, so we're still showing that here. Okay, and then the last phase then, cytokinesis 2, is when the cytoplasm splits. So we're going to end up with 4 cells. So, in the end, we're getting 4 cells. And they are haploid cells. So you'll notice they have half of the DNA as the original cell had. The original cell looked like this. This is the parent cell, right? Then it divided. And it divided again, and now we have half of the DNA. Technically, I'm showing here the parent cell after the DNA has been divided. So before DNA makes a copy of itself, the parent cell would really look like this, which is twice of the DNA that these have. Right? So these 4 are the daughter cells. These are sperm cells, or they're egg cells. If they're egg cells, they don't all develop into an egg cell. Only one of the 4 actually does. But if they're sperm cells, they all develop into sperm cells. But I want you to notice the difference between the daughter cells and the parent. Not just the amount of DNA, but the fact that it's mixed. And these daughter cells have half the DNA as the parent cell. And not only that, they're all different. So these actually would be different. all of them. And this shows that there's even more mixing happening. So there's more shuffling going on. So there's shuffling happening if there's crossing over, but there's also shuffling happening when the eggs and sperm are actually made. And then during fertilization, there's even more shuffling. So the fact that there's all this shuffling happening, this ensures that there's a lot of genetic variation. In other words, there's a lot of differences in the genes. So this is what makes populations genetically diverse. And the more genetically diverse they are, the stronger they are, because they're going to be some individuals that are resistant to diseases and others won't. If we were all the same, we would have the same immune system. And if you think of like COVID-19 now, some people are more susceptible than others. If we all had the same immune system and that immune system was susceptible to something like COVID-19, it would wipe out the whole population. But genetic variability is what keeps populations alive. And that is really due to the process of meiosis and sexual reproduction. And that ensures that the population is strong because it has a lot of genetic variation. The more genetic variation, the stronger the population is. And this is why even endangered species or species that have very small population sizes, they have low genetic variability. So even if we were to breed them and make more, the genetic variation is still limited. So they're really like inbreds. And this is a problem with a lot of artificially bred animals like dog breeds. This is why a lot of them have health problems. They're so inbred. There's such limited variation among the genes. Wolves have a stronger population. So wolves do not get a lot of the diseases that our dog breeds get, even though they all really came from the wolf. But we've bred the wolf to look different ways. So we've bred out these breeds of dogs because we like the way they look, or whatever. And then when we did that, though, we reduced the size of the gene pool. So we reduced the genetic variation. And there are consequences to that. So that's why a lot of dog breeds, their lifespan is short, and they do develop a lot of health problems, the purebreds, because they're basically inbreds of each other. There's not much genetic variation. So genetic variability is very important if a population is to survive. And it comes from meiosis and sexual reproduction. So it's awesome. Science rocks. Woo!